Next up, we have Professor Jerry Moeller. Jerry is a professor in the departments of psychiatry, pharmacology, and toxicology, and neurology at I think Brian's going to get your headset on.
Should I just use the regular one?
Yeah.
Okay. I'm not a big walker. Okay, well, first I wanted to thank Mike Friedlander and Warren Bickel for inviting me to participate in the conference. And the time I have left between you and lunch, what I thought I would do is just provide an overview of the translational research in the neurobiology of addictions that we've been doing at VCU with a focus on impulsivity and cue reactivity. So it's sort of a nice segue from what Warren was talking about. So just to give you a background of where impulsivity fits in the overall model of addiction. So this is one of the papers published by George Koob and Nora Volkow, this time with Tom McClellan this year, providing an overview of the neurobiology of addictions. And what they have in their model is different phases of addictions and the brain circuits that they believe are associated with those phases. So the initial phase is the binge or intoxication phase, which is focused on reward. So the rewarding effects of the drug really are primary in that phase. Then that's followed by a withdrawal and negative affect phase where the acute effects of the drugs have worn off and now the behavior is driven by the withdrawal and avoiding the negative aspects. And then finally a preoccupation and anticipation phase where the drug cues and craving really drive the substance use. And where this fits in is that where impulsivity and cue reactivity fit into this, besides the cue reactivity down here, is so that you're moving from a voluntary action phase of substance use where you have abstinence or use that's constrained through periods of time where you go through from sometimes taking the drug without more than you would like or using more than you would appreciate, and to the point where you have impulsive action, relapse, and compulsive drug use. So it goes from voluntary use to this phase, which is really where the addiction comes in. So what I would like to give you an overview of is the evidence we have from translational research that impulsivity and drug cues are related, and drug cue reactivity are related, and that they could be a potential target for medication development for addictions. Sorry. Oh, sorry. I'll get closer again. So I'm going to share with you some behavioral data, both from preclinical and clinical behavioral data that we have, as well as some brain imaging and neurobiology data from rodent brains, and neurotransmitter data, to ultimately looking at pharmacotherapy, potential pharmacotherapy for the disorder. So to start with in the behavioral data, so as Warren talked about, there are different behavioral lab measures that have been used as measures of impulsivity. One of the measures we've been using is it's a variant of a continuous performance task. So the CPT is commonly used in kids with ADHD. This is a more complex version of a CPT. And the way this works, so this is the immediate memory task. It was developed by Don Doherty when he was at UT Houston. And the way this task works is that the participants are asked to press a button every time they see a number that matches on the screen. So there are a series of 5-digit numbers that are presented to them on a computer screen. They're on for half a second and off for half a second. And every time they match, they press the button. So, and it's easy to pick up on when the numbers match because besides the matching, there are periods of time where they don't match at all. And if you're paying attention at all, you can pick up on the numbers match or they don't match. But rarely, there's a presentation of a stimulus where the number matches just by 1 digit. And an error on this kind of trial is a commission error, and that's a measure of impulsivity. What we've seen and others have seen is that populations that have trouble with impulse control have a hard time inhibiting a response for that kind of stimulus. And this is just some data showing that groups with high impulsivity, so in this case it's cocaine-dependent individuals. This is a paper we published in 2005, but repeatedly in different populations of impulsive individuals, borderline personality disorder, other kids with conduct disorder have higher commission errors on this task compared to healthy controls. And this parallels their self-reported impulsivity as measured by the Barrett Impulsiveness Scale. So the way we're measuring drug cues in humans is a modification of the Stroop task. So if you're familiar with the Stroop task, where the participants are told that they are to press a button when they're looking at a computer screen based on the color of the word, and ignore the meaning of the word. And the way this task differs from the normal Stroop task is that alternating between household words are drug-related words. And what the attentional bias is a phenomenon where drug-dependent individuals, when they see drug-related words, have a slower reaction time for those words compared to the neutral words. And so this was a paper published in 2011 where showing cocaine-dependent individuals had this attentional bias or slower reaction time to the drug-related words compared to the neutral words. So showing that they have a higher cue reactivity as measured by this task. What was interesting is that the performance on the immediate memory task and the drug cue reactivity on the cocaine Stroop task was related. So individuals who have higher attentional bias, so they have more of those slower reaction time for the cocaine words compared to the neutral words, had more commission errors on the immediate memory task. So suggesting there may be some relationship with this. Obviously, this is just a correlation, and we can't look at that more than in the correlation except with other methods that we're using. So the translational sort of back translation that we've been doing with a collaboration with the University of Texas Medical Branch with Kathy Cunningham and her group uses tasks that we've selected to be translational to the human task. So the task we're using for impulsivity is the one-choice serial reaction time task. So this is a modification of the standard 5-choice task, and it was modified because this task takes less time to train the rodents in compared to the standard 5-choice task. But we get very similar results. The way this task works is that the rodents are trained through operant methods to, when a light comes on, there are 5 holes for nose pokes, and when the light comes on, they're trained to poke their nose into the hole where the light came on, after the light came on. And if they do that correctly, they are rewarded with a food pellet. Other things that they can do is if they respond to the wrong hole where the light didn't come on, the house light comes on and they are not rewarded, so they don't get any reward for that. Other kinds of behavior we see would be omission error, similar to what we've seen with a continuous performance test. They are not rewarded for that. And the error that we're interested in is this premature error, so this commission error where they respond before the light comes on. So it's the right stimulus, but they do it before the light comes on. And if you use this task in rodents, you can separate out rodents with high impulsivity and low impulsivity. So, and this is fairly stable over time. So you can see that they just separate rodents into higher versus lower premature responding. And we do this over time, and this is just with standard Sprague Dawley rats. You can generate a group that are high impulsivity versus low impulsivity. So the cue reactivity measure is based on drug self-administration. And this is the model that Kathy's been using. So the rats undergo surgery to insert an intravenous catheter for cocaine and self-administration. They're then trained to self-administer cocaine, which they do. And at the same time they're administered cocaine, there's a stimulus, a light comes on, so that's paired with the cocaine administration. They then undergo a period of forced abstinence. And after the period of forced abstinence, they are reshown the light, the stimulus, and without cocaine, and we measure the amount of responding on that lever press that they have for just the light without the cocaine, and that's the measure of cue reactivity. So an interesting back translational issue is sometimes, often, preclinical researchers had used extinction to look at this. And we told our preclinical colleagues that clinically we don't do any extinction with our cocaine users. They have forced abstinence sometimes, usually in jail, but we don't have extinction. So we said if you want to really be translational, you should do that with the forced abstinence. So, and this is the similar behavioral findings that we had in humans. So the rats that were initially trained on the one-choice serial reaction time task, separated into high and low impulsive groups. They then undergo cocaine self-administration, and then have the cue reactivity measured by the task, which I just showed you. The high impulsive group had a significantly higher cue reactivity compared to the low impulsive group. So it parallels what we're seeing in the human research studies. So that might be interesting behaviorally, but so what's behind that? So one of the ways we're trying to look at that is looking at the brain regions that are associated with this behavior. And in humans, what we've been using is looking at effective connectivity using fMRI and dynamic causal modeling. I know you guys are doing that here as well. So this is a paper we published, I think it was last year, looking at inhibitory control in cocaine users and controls, looking at the effective connectivity. The tasks that we've been using in the scanner is a go, no-go task, which is probably the most commonly used inhibitory control task in the scanner. And the way this works is that the participants are told to press a button when the stimuli match exactly, and to withhold a response if they don't. And most of the time they do match exactly, so it builds up a prepotent response. And intermittently, there's an easy no-go session where the lines are parallel and diagonal, so it's pretty easy to detect the difference. And then there's two diagonal but going opposite directions, which is a little harder to detect and to make more errors. And so what we found is, so again, with effective connectivity, dynamic causal modeling, you're able to look at the directionality of the influence of one brain region over another. So as opposed to standard functional connectivity, you're just looking at correlations. This actually looks at what brain regions are driving activation in the other brain region. And what we found was the anterior cingulate, during the hard no-go task, when the cocaine users were able to effectively inhibit a response on the go, no-go task, that reduced the effective connectivity between the anterior cingulate and the caudate. There was no effect of this on healthy controls, showing there's something different going on in the brain circuitry during successful no-go response in the cocaine users. It also showed that the anterior cingulate was important for the successful inhibition. During the way we're looking at cue reactivity in the scanner is using that same attentional bias task, the cocaine Stroop task in the scanner. So I just showed you earlier. And what we've seen during the task is that the drug cues compared to the neutral cues enhance the connectivity between the insula and the anterior cingulate, and also between the anterior cingulate and the medial orbital frontal cortex. So the drug cues are having an effect driving the anterior cingulate and the anterior cingulate of the medial orbital frontal cortex. So suggesting, if I was looking for a reason, a brain region to study for where the overlap is between impulsivity and cue reactivity, the anterior cingulate would be a good place to start. So just in summarizing this, the reduced anterior cingulate to caudate effective connectivity in cocaine users was driven by applied differences in top-down control in the cocaine users compared to the controls. The cocaine cues enhance that connectivity between the insula and ACC. So the ACC is a key brain region for both both impulsivity and drug cue reactivity. So if we go back to this, and I borrowed this from Drs. Volkow and Koob, so to give them credit for this in case there are any copyright issues down the road. What we're seeing is for the go, no-go task, the anterior cingulate there's a reduction in the drive of the anterior cingulate to the caudate. And for the cue reactivity task, we're seeing an increased activity between the insula and anterior cingulate, and between anterior cingulate and orbital frontal cortex. So it's suggesting here that, again, the ACC is a place to look at for this. So could the ACC, if we go there and look for differences in neurotransmitter function, could that be a way for us to look at potential pharmacotherapy targets? So that's the next part of what I was going to talk about. So this is a paper published in 2014 where looking at the medial prefrontal cortex, and this would be something that would be interesting for a discussion in the room. Obviously, the rodent prefrontal cortex is very different from the human prefrontal cortex, and we have discussions all the time about, well, what is the best area if you want to look at for the anterior cingulate cortex or the dorsolateral prefrontal cortex? And they'll say, well, some papers say it's this, and other papers say it's that. But at least based on some of the papers, this area that we examined in the medial prefrontal cortex in the rodent was analogous to the included areas that are analogous to the anterior cingulate in humans. And what we've seen is that the high impulsive rats on that one choice reaction time task had lower medial prefrontal cortex serotonin 2C receptor levels compared to the low impulsive rats, and that there was an inverse correlation between the medial prefrontal serotonin 2C receptor levels and premature responding on the one choice. So suggesting that serotonin 2C receptor function was important. So that ties in with where we can go from here, and it's interesting listening to the industry discussion earlier. So one of the big issues that we've had for many, many years trying to study serotonergic function in humans and looking at ways to manipulate serotonin function in humans is that we have very few compounds available to us that are selective for serotonergic receptor function until recently. So one of the compounds that was talked about in the earlier session is pimavanserin, is a selective 2A receptor antagonist. And recently, not maybe I guess a year and a half or 2 years ago, lorcaserin was approved for obesity. So you might say, well, that's not talking about what is a CNS, is that a CNS approval for a drug? Well, I think it's working through the CNS to have its effect on appetite. So we are looking at lorcaserin now as a potential treatment for addiction. So this just shows you that where the 2C agonists, based on a number of different studies and different 2C agonists, support that it looks like this is a potential mechanism to treat both impulsive action as well as cue reactivity and drug self-administration. So now there's some interesting data coming out of Kathy Cunningham's lab as well as other labs that not only do the 2C agonists reduce self-administration and cue reactivity for cocaine, but they also reduce self-administration for nicotine and most recently for oxycodone. So it's a potential avenue for treatment of drug addictions that cut across disorders. As Warren was mentioning earlier, it's not specific for a specific substance of abuse. So this suggests 2C receptor agonists could be a target for medication development for addictions. So in summary, I think hope I've given you some evidence that impulsivity and drug cue reactivity are related in both preclinical and clinical studies, that brain imaging studies suggest overlapping neurocircuitry for these two, and that there is data that 2C receptor agonists, and interestingly, I didn't just talk about this, 2A receptor antagonists have similar effects on reductions of both impulsivity and drug cue reactivity. So suggesting that translational research may be a way to move forward for medication development for addictions. So NIDA, unlike some of the other institutes, is very interested in medication development, largely because most pharmaceutical companies are not interested in drug abuse as a target for medication development. So they've been very supportive of our research. So obviously this is a a large research team, some of whom I moved up with me to VCU from Houston, and some who stayed in Houston and also in Galveston. So Kathy Cunningham and Noel Anastasio do all of our preclinical work at University of Texas Medical Branch. We have fellows and behavioral research collaborators in Houston as well as here, and a large group of brain imaging research collaborators led by Joel Steinberg and Young Swo Ma. And we've been fortunate to be funded by the National Institute on Drug Abuse. So with that, I'll open up for questions.